Hello, this is Giovanni Campanile, MD, from the Sicilian Secret Diet Plan, and Sandra and I today are thrilled to be having a chat and interview with Professor Jed Fahey. Jed is a world-renowned expert on the benefits of sulforaphane and other components found in broccoli and broccoli sprouts. He's a retired professor from Johns Hopkins University and clinical biochemist, and he's one of the principal investigators discovering that broccoli sprouts contain high concentrations of the precursor to sulforaphane, glucoraphanin, and all of the many health benefits originating from these compounds. Since his initial discovery, he has made momentous contributions in this field, discovering the benefits of sulforaphane on cancer, heart disease, brain disease, on the microbiome, autism, and many other illnesses. Today, we're going to have a fascinating discussion about this remarkable compounds that are found in these plants. Initially, we'll talk about how broccoli was brought to the United States by the Dorego brothers from Sicily. And then we'll talk about Jed's path from his early days to discovering many of these earth-shattering discoveries. One of the many publications that he has done over his career was published in the Proceedings of the National Academy of Sciences, which show that small quantities of cruciferous sprouts may protect against the risk of cancer as effectively as larger quantities of mature vegetables of the same variety, which is quite fascinating. Also, he has discovered that Moringa, which is a tropical plant, has a high concentration of similar isothiocyanates, moringin, which, like sulforaphane, protects against cancer, heart disease, and diabetes. We'll also talk about the NRF2 pathway, which is a nuclear transcription factor that is activated by sulforaphane. And this leads to the production of cellular proteins, which then lead to antioxidants and detoxification. So these are cellular proteins that are activated after we ingest these compounds like sulforaphane. In addition, we'll talk about the 10 enzymatic steps in the production of glutathione, which is the most prevalent and powerful antioxidant in our body. We'll also talk about how sulforaphane sometimes is misclassified as an antioxidant because it's truly not an antioxidant in itself. It's an indirect antioxidant in that it upregulates many cytoprotective genes. And it's also involved with other things such as heme and iron metabolism, lipid metabolism, and antioxidant enzymes. And the NRF2 also stimulates protein enzyme production and other factors which are helpful for protection against lung disease, arthritis, hypertension, also protection against H. pylori infection of the stomach, which contributes to gastric irritation, gastroesophageal reflux, and also gastric cancer. Broccoli sprouts have about 10 to 100 times the sulforaphane compared to broccoli, and we'll talk about how broccoli sprouts themselves are incredible from a nutritional standpoint, and at the end, we'll also add a link to how to make your own broccoli sprouts. Also, we'll talk about how much you should consume. 2 servings daily are shown to be associated with a reduced risk of colon cancer and breast cancer. Also, supplements that may contain glucoraphanin and may be helpful if you cannot consume broccoli or broccoli sprouts. Also, we'll talk about ischemic protection from reperfusion. This is an important concept because when we get blocked arteries, especially in the heart, and then the arteries are open, either with a stent or with thrombolysis, there can be injury, oxidant injury, and this sulforaphane helps protect against that. We'll also talk about the effect on heat shock proteins in sulforaphane in the disease progeria, which is an accelerated aging disease. And at the end, we'll also have some links to specific seeds that you can purchase from Amazon or from johnnyseeds.com. And also, we'll talk a little bit about Jed's involvement with companies like foodnerd.com. So without any further ado, we'll get on to this exciting and fascinating discussion with Jed. Well, thanks again, Jed. This is great. We're very excited to talk to you. Sandra and I are both, big fans of your work. You've done some amazing work that's affected, so many people.
And has affected our behavior because we have been eating more broccoli sprouts and consistently making it. So
I just wanted to bring our broccoli sprouts that we make regularly. Well, nice job with them, I can see. And I'm very much a fan of making your own rather than buying them in the store, because they sit on the shelves in the produce select section for quite a while sometimes, and they get pretty funky.
Yeah, I've seen that. Even in good stores, they're moldy. Yep. You can't. It's hard to keep them not getting moldy even at home. So I can't imagine in a supermarket how they can do it.
And then we'll talk. I guess you'll tell us what is the exact and perfect time to eat them. How much can we, for how long can we store them? I guess you'll guide us, and we'll guide our listener through the making and storing of the broccoli sprouts.
But before we get into that, let's get into your history. The whole discovery process of, the glucoferrafenin, the myrosinase, the sulforaphane, your whole backstory. We'd love to hear that, and our listeners would love to hear it also.
How far back should I go? Not to the womb, I assume.
Well, maybe if your mother was eating broccoli sprouts, yes.
So, all right, well, we'll go pretty far back then. My mother's the descendant of a couple of Italian immigrants. Campisi was her maiden name, and she was a wonderful cook, and we ate plenty of broccoli when I was growing up, as well as other vegetables, and good Sicilian cuisine.
And actually, I have to say that broccoli were brought to the United States by two Sicilian from Messina's, two brothers.
The Dorigo brothers?
I'm sorry?
Was it the Dorigo brothers?
The Dorigo brothers. They brought the broccoli first in California, and then it became a success. But actually, before him was actually Thomas Jefferson that brought some broccoli and started planting them, but they were considered very exotic.
I remember reading about that, and in fact, I think they even say something about it at Monticello and one of the displays about the garden diaries that he kept. He
had several kinds. He had white, he had purple.
Yep. So I was eating broccoli when I was very young, and anyway, I was going to be an aquaculturist, a marine biologist growing up, or a concert cellist, and I had an accident, dropped an air conditioner on my middle finger of my left hand, just entering audition season. So I decided to go to college, and I studied science instead, and so that's sort of the way my career evolved, not in a very structured way, but so I studied science. I like marine biology, grew up near the water, and then studying algae in the 70s. There was no market, there were no jobs in microalgae. So long story short, I spent a fair amount of time in the ag biotech industry as a plant guy. I was welcome there. And then I went to Johns Hopkins in 1993 because Paul Talalay, very distinguished senior scientist, had just discovered sulforaphane in broccoli in 1992, and he was looking for someone with botanical chops, so to speak, to help find better broccoli. I quickly, I took it as a challenge. I was tired with working in the ag biotech industry at the time. There was a lot of promise, a lot of hype, and nothing really satisfying to me. Let's put it that way. So I took Paul's challenge on. I remember distinctly, he said, when he was interviewing me, he said, I'm 70 years old. You sure you want to come to work for a 70-year-old man? He passed away 3 years ago at age, or was it a few years ago, at age 95, and was working essentially the whole time. So we had a quarter of a century together. And
broccoli sprouts did help him.
Yeah. I don't know how many sprouts he ate. I know he ate broccoli, but yeah, I could not work on broccoli in the middle of Baltimore, all year long without importing it from all over. I wound up growing the seeds in growth chambers in my lab up in the middle of East Baltimore, and quickly discovered that broccoli sprouts were much more potent sources of this compound sulforaphane that he and Yushin Zhang, his medical student, had discovered. And Paul and I decided to commercialize, to try to promote broccoli sprouts, first of all, because they were green, they were plants, they were rich in this phytochemical, and we thought that this might be something that more than just a handful of Americans would be interested in, eating sprouts, that is. Whether they were or not is sort of besides the point now, because what happened is there were some sprout safety issues that the FDA was all over back in the late 90s when we decided to promote sprouts. We started a company to do that, and that company very quickly wound up having to pivot to making supplements, broccoli seed extracts that are used in some of the best supplements on the market right now. But so we got uninvolved with growing sprouts commercially very quickly, and Paul and I actually divorced ourselves from the company so that we could continue to do clinical studies with broccoli and broccoli sprouts and sulforaphane and its precursor glucoraphanin. And anyway, let me stop talking for a second, but that was the broccoli story in a nutshell. And then for 27 years, I've been spending a lot of my time with broccoli sprouts, sulforaphane, as well as with a tropical plant, as well as with a lot of other phytochemicals, but with a tropical plant that has very similar activity called Moringa.
It's also a fascinating plant. Yeah, and so I guess you'll tell us more about it. But now it seems that this magic component has kept you interested up for a very, very long time. So let's tell us, all our listeners, why did it keep you interested for so long, for 25 years? What is it about it?
What is it about it? Well, again, in a nutshell, broccoli and broccoli sprouts produce a compound, produce obviously a lot of phytochemicals. I'm a huge fan of phytochemicals. I'm a huge anti-fan of ultra-processed foods. For a lot of reasons I'd love to talk to you about if we have time. But broccoli, the phytochemical that Paul and Yushin discovered in 1992 called sulforaphane it turns out, and this is the work of not just our group, but many others around the world now, it turns out it is effective biochemically, metabolically on a number of very important pathways, from immune response activation to being anti-inflammatory to being what we call an indirect antioxidant. I can come back to that. To being helpful in animal models anyway in wound responses, being important in cell cycle arrest and apoptosis, which is important in cancer, getting rid of precancerous or cancer cells, among other things. It is selectively antibiotic. We have done a number of studies with Helicobacter pylori, and it's turning out it's also antiviral. Colleagues at Hopkins, after I left, have looked at its antiviral activity against the common cold virus and against SARS-CoV-2, the causative agent of COVID. And it's involved in the heat shock response. So, those are just sort of a few things that it's involved with. The pathway that we keyed upon, and many others have also, is related to the antioxidant and anti-inflammatory response, and that's the so-called NRF2 or NRF2 keep one mediated response. Without I have to get a little technical. That's the purpose of the podcast, right? But so NRF2 is a nuclear transcription factor that sulforaphane activates. I can describe the way it does that, but for now, let's just say it activates this pathway. And there's an
intracellular activation, correct?
Right. So once sulforaphane is absorbed into the bloodstream, into the cells, I should say, there is I get a little more technical then, there's a chaperone protein in the cytoplasm of the cell called keep1. It's actually a dimer, 2 molecules. And it holds, if you will, I mean, have to use sort of cartoons as all the scientific papers that describe it do. So this dimer holds something called NRF2, which is a nuclear transcription factor. And when sulforaphane enters the cell, it attaches to 2 cysteine residues on this tether protein. It changes the conformation, lets NRF2 travel then, and it goes into the nucleus, and it sits on an antioxidant response element on the DNA, and permits the transcription of a variety of genes, a whole suite of genes, and the estimates are that between 3 and 5% of cellular protein is governed or controlled in one way or another by this NRF2 factor. So it's very important, and it turns out that many of these proteins that are upregulated have to do with detoxification. As I say, antioxidant response, an anti-inflammatory response, detoxification of things like AGEs, air pollutants, and so on, mitochondrial support. So it's a very central.
So it helps in detoxification, also helps in glutathione synthesis. I've read some studies showing that brain glutathione levels can actually go up with sulforaphane, and also with NADPH synthesis. There's a whole movement now to try to increase NADPH with other modalities. I don't know how robust is that.
You want to say what that is?
Well, NAD and ADPH is a molecule that's very important for cellular and mitochondrial function. And some people take NMN or NR to boost this, but sulforaphane actually boosts this also. So I'd like to hear what you have to say about that.
Yeah, it does. We haven't studied that pathway very much, frankly. So I'm not going to comment on NMR versus other modalities of supplementation. But you mentioned the glutathione pathway. So absolutely, one of the glutathione pathway, which involves recycling oxidized glutathione, as well as making it de novo, has, I don't know how many, a number of steps, 10 or 15 enzymatic steps directly involved in it. And so this NRF2, the NRF2 response upregulates some of the key sort of roadblock enzymatic steps in that pathway. allowing glutathione to be both made, more of it to be made, and for it to be recycled. And this becomes particularly important when you think about the fact that glutathione is the body's most prevalent antioxidant. It's one of our most important native endogenous antioxidants. It's present at millimolar levels in the macula of the eye, for example, and it's used throughout the body, the nervous system, as you mentioned. So,
correct.
As an antioxidant, I'm sorry, what?
We lose it with age, correct? And that is what partly the problem is of aging, losing. Go ahead.
Right. We lose a lot. We lose a lot with age, but we can keep a lot of those pathways at least more robust by exercise and proper diet and proper sleep. I mean, it's a pretty simple trifecta when you think about it. And
just for our listeners, the reactive oxygen species are created as part of metabolic byproducts throughout our cellular, throughout our bodies. So this is why glutathione and the antioxidants are so important. I just wanted to point that out.
Absolutely. So we live in an aerobic environment, meaning we breathe oxygen, and that's a wonderful thing for all of us. It keeps us alive, but there are downsides to it. And there is, as you say, oxidation of macromolecules throughout our body. And we're constantly neutralizing that oxidation because otherwise we would rust away, right? I mean, oxidation is when iron oxidizes, it forms rust. And it's I think, a good analogy for the human body. We get pretty decrepit pretty fast if we didn't have mechanisms for, if we didn't have antioxidant mechanisms. One fallacy that I'd like to dispel before sort of preventively or prophylactically is that sulforaphane is an antioxidant. So it's not actually a direct antioxidant. And chemically, it does not act as an antioxidant, meaning it's not something like vitamin C that will sacrificially react with compounds to neutralize them. We call it, and others now have adopted this phraseology, we call it an indirect antioxidant because it upregulates so many antioxidant pathways and enzymes in the body.
In addition to NFR2, and it also upregulates other pathways like mTOR and FOXO and those pathways.
Not all of them, but I would say it's the NRF2 upregulation that's key to this antioxidant response because of all of the genes that it regulates. I have someplace, I'm not going to go searching for it now, but I've got a slide. I've got a list that I took from someone else's paper actually, and in fact, I'm looking at it now for you, but. If you look at all of the known proteins, which for which the genes are upregulated by NRF2, they include, for example, biotransformation and detoxification genes. I'm looking at my short list now. I don't see FOXO on here. I haven't seen reference to that being upregulated, but enzymes involved in heme and iron metabolism. Enzymes involved in lipid metabolism, as we discussed, antioxidant enzymes, and when you think about indirect antioxidants, and again, I like to compare it to vitamin C. If you take a bolus of vitamin C orally, and then you look in the blood, it spikes really quickly, and it comes down really quickly, and it's gone. So unless you mainline vitamin C, and unfortunately there are people who there are all sorts of wacky ideas about vitamin C. So there are people who want to take it constantly in mega doses. Nobel Prize winner Linus Pauling was one of them. But anyway, if you do what most people that take vitamin C do, it goes up, it comes right back down, it's gone. The excess is peed out, finito. The next day, you spike it again. If you do that with sulforaphane and follow the pharmacokinetics, the same thing happens. It spikes, and then it disappears from the blood. But what happens is it upregulates this 3 to 5% of the genes. It upregulates this large number of cytoprotective genes, and the enzymes that are formed. These are genes for proteins. Proteins or enzymes are proteins. And as you know, enzymes have some half-life, so they last for a while, they do their thing, and eventually they're degraded or they cease functioning, and then they're chopped up and the amino acids are reused. The half-lives of the NRF2-induced enzymes that we've discussed are, by and large, from 5, 6, 7, 8 hours to days, and some of them even a few weeks. So what you're doing when you get that bolus of sulforaphane is goes into the cells, turns on the NRF2 mechanism as we discussed. The enzymes are created, they function for hours to days. So
there's a robust, prolonged effect, despite the fact that it's not in your system any longer.
Exactly. And so you don't need to have it on board. So if you use the traditional pharmacologic paradigm, area under the curve or peak plasma concentration or any of those things, they're not really that relevant. But they're not really relevant at all, other than the fact that you want enough to create this NRF2 response. Now, if you look at other mechanisms of sulforaphane's action, like, for example, the antibiotic effect against Helicobacter, that's a completely different ballgame, we think, and that's because there you're asking the molecule itself to exert its antibacterial effect on the critters that are swimming around the mucus layer of your stomach. So it's very much more a direct.
That's a fascinating, that part is, I want to get into that a little bit more, but just to go back a little bit. Now, I'm fascinated by the fact that our gut microbiome, and we focus in on gut microbiome very much in our book and in our patients and in our practice. And, the fact that our gut microbiome, the friendly bugs, produce the enzyme that converts the glucoraphanin into sulforaphane, which, to me, that's amazing. That it sounds to me that we're meant to eat this food since our good bugs have this enzyme. But that enzyme is also in the plant itself, correct?
Yes. So the enzyme myrosinase, as you point out, is both in the plants and in our bodies. Interestingly, it is not in our mammalian bodies. So if you were bacteria-free, you would not have that enzyme. The reason it's present in our body, so it's not present in the cells of your bodies. It's present in your gut. Primarily, we think the large intestine, but also perhaps in some of the bacteria in the small intestine, especially the lower small intestine. Why did we evolve a population of bacteria, a microbiome that contains this enzyme? We can and should speculate. Is it more prevalent in Sicilians and Mediterranean Europeans that historically ate all sorts of wild cruciferous plants? I don't know. I'm not aware that anybody has studied that with enough rigor to say anything conclusive. Is it present in Inuit populations that eat whale meat and never see a crucifer? I don't know. I don't know if that's been examined, but this is all really fascinating. Or is myrosinase, as we call it in bacteria, myrosinase is a thioglucosidase. It's very similar to a beta glucosidase. So do the bacteria which contain it have this enzyme for a different purpose? Do they do something different with it? And when you eat cruciferous plants, hey, we can break this down too. We don't let us.
Small interruption to let the dog in.
Of course.
Sorry about that.
So, this is, again, like you said, fascinating, interesting to figure out how did this happen? Why did it happen? We know we've been eating broccoli since the Romans' time, so probably we have accustomed to digest it. But what I'm also very curious about is the antiviral, antibacterial effect of the sulforaphane. Is it independently from the plant broccoli? It's just the isolated? Because I know broccoli has a lot of other important aspects, has elevated vitamin C, 150, I think, percent of the amount of what is recommended to take daily, has calcium, close to milk levels. So is the plant, eating broccoli, the whole plant that has the overall effect, or is just the sulforaphane, isolated?
Well, we, as laboratory and clinical scientists, we're faced with this dilemma when we try to study diets and plant food, that if we're trying to relate how much of an effect we're seeing to the amount of a particular compound of interest, in this case, we've talked about it already, it's very difficult to do, especially with free-living people that are getting a diet, whose diet you can't control beautifully. So the short answer to your question is sulforaphane is the active moiety when it comes to the antibacterial activity and in fact, the antiviral activity that we're talking about. But the first clinical trial that we did with Helicobacter pylori was in Japan, and subjects were fed for 8 weeks daily fresh broccoli sprouts. So the effect we saw was from broccoli. We believe it was due to the sulforaphane for many reasons, because all the lab tests and the culture tests used pure sulforaphane. And in fact, there was a small study using broccoli sprouts before us. This is based on our work, but this was done by Ann Silverman in Detroit, and same thing. She fed broccoli sprouts to people. So, yeah, I mean, it's a direct effect of sulforaphane, but it can be elicited by feeding the plant that contains sulforaphane.
We have fibers in the plant too, so you would expect that the microbiome gets excited about that and maybe utilizes a little bit better. So there might be some synergistic effects.
Right. I should point out one of the things we suspect, but we haven't proven, is that in addition to the direct antibacterial effect on Helicobacter, if there's going to be any sustained control of it, it's likely that, we're talking about acting in the stomach, high up, essentially the first thing the food hits, but then hours later, having glucoraphanin acted upon in the large intestine, having sulforaphane produced, absorbed, and perhaps there's an effect in the stomach epithelial cells that also inhibits lesion formation, inhibits some of the things that Helicobacter does in the stomach. I like to go back to that evolutionary or the diet question for a minute. We keep in this conversation talking about broccoli. We all love broccoli, yes, but it's important to realize that there are, I believe, 16 families of plants besides the Cruciferous family that contain glucosinolates and myrosinase and isothiocyanates. And so these include many of the, your Mediterranean diet plants. I mean, things like arugula, rocket, kale, etc. Those are all cruciferous vegetables. But there are, as I say, there's 16 in total families. So there are 15 other families, most of which are far less well populated by edible species. But for example, the caper family, you're familiar with capers, of course, that contains this, and the Moringa family, which I mentioned Moringa earlier. So all of these other plants contain compounds very similar to glucoraphanin and sulforaphane. A few of them actually also contain this pair, glucoraphanin and sulforaphane, but none of them in nearly the levels that broccoli has. So, it's important to realize that broccoli is essentially the only cruciferous vegetable that delivers sulforaphane. You can't say eat cruciferous vegetables, they all contain sulforaphane, but you can certainly say eat cruciferous vegetables, they all have very beneficial glucosinolates, isothiocyanates. These are good for you, because the fact of the matter is
for our listeners, what you just mentioned, they're all good antioxidants.
Indeed, and there are something like 120 different glucosinolates. Many of them have very similar modes of action to glucoraphanin and sulforaphane, and some of them different modes of action. In different assays, some may even be more active, some less active. We think sulforaphane sits at the top of that pyramid as sort of the king or queen of the roost, but that's not to besmirch the benefit of eating other plants that contain this suite of phytochemicals.
And what about broccoli sprouts in comparison to broccoli? As I showed in the beginning, this is my broccoli sprout. I actually have a lot of my patients make broccoli sprouts because it's so easy to do. We make it frequently. It's part of our routine to make it, and there's really no big effort in making it, and it's such a nice thing to add to your salad. So what is the difference between, and also I'd like to know the Moringa too, but what's the concentration in broccoli sprouts versus broccoli versus Moringa, that kind of thing? So the concentration in broccoli sprouts are sort of been saying this for years, but it's a fuzzy number, but it's somewhere between 10 and 100 times the concentration in market stage broccoli. And I have to say it's fuzzy because there is no standard number for broccoli. As you know, when you go to the grocery store, even if you go to a farmer's market, most of the time you don't have any idea even what cultivar of broccoli you're buying. So you're buying broccoli. It's labeled broccoli with a big pink rubber band or purple rubber band. Or if you're at the farmer's market, if you're lucky, you can get the farmer to tell you what cultivar he or she grew. But typically, the American consumer doesn't know what they're getting. And we've studied probably hundreds of different random accessions pulled from grocery stores, and there's absolutely no way you can tell by looking at that broccoli how much glucoraphanin is in it. Color, smell, quality, lousy, abused, thrown around the produce section, dried out. You really can't tell without doing a chemical analysis. So, but if we average all of the heads that we've had the pleasure to analyze, and take an average number, and then do the same with, say, 3-day-old broccoli sprouts grown from a variety of different seeds, it's between 10 and 100 times more. Wow.
Now, the thing about, I've told you what the problem is with traditional broccoli heads. You just can't tell. The sprouts, though, theoretically, you can tell, because when you think about how you make sprouts, you hopefully sanitize the seeds with Clorox or something else. You rinse them, you stick them in a jar, and you rinse them and talk to them, 3 times a day. And 3 days later, you put a cap on them and throw them in the refrigerator and eat them for the next week. if you knew the potency of the seeds you started with, then you could fix a number to the glucosinolate content or the sulforaphane content, if you will, of those sprouts much more easily. So, for example, when the company that Paul and I started with his son Tony, who's still running it, when they make extracts of broccoli seeds, they have developed varieties of broccoli seeds, or they've stabilized really varieties of broccoli seeds that have a known concentration of glucoraphanin. They analyze those seeds regularly coming in from the field, and then obviously when the extracts are made and produced, the normal consumer, including myself, and I assume you, does not buy seeds by glucoraphanin titer. They buy them. There are two ways to buy. There are two things you think about when you buy seeds for sprouting. One is, are they organic? Are they not coated with pesticides? And the second is, do they have a pretty good germination rate? In other words, is it going to be like cruddy popcorn where there are a bunch of unpopped kernels, or do most of them sprout? Other than that, I'm sorry, there's a third thing, price. Because hybrid broccoli seed, which is used to grow the broccoli you buy in the grocery store, is very expensive. I don't know what it costs nowadays, but upwards of $40, $50 a pound would be my guess. The seed one buys for making broccoli sprouts is typically something called open pollinated, or some old variety where that's not planted much anymore because there's too much variability in the field. It's hard to harvest it all at one time with a machine. But that broccoli is much cheaper, and those are the seeds you typically buy to sprout. So at any rate, there's a long way of saying it's very hard to tell what that differential is between sprouts and market stage, but it's 10 to 100-fold.
And what about the seeds themselves? Don't the seeds themselves have glucoraphanin in them? You could consume the seeds themselves, correct?
You could. The seeds themselves are bitter, not very tasty at all. If you roast them, they get a sort of nutty taste, but you have to be extraordinarily careful roasting them because there's a very fine line between that nutty taste and scorching them and burning them. And for people to eat large quantities of the seeds, the seeds are 30% fat or so, 30% lipids. There are fats in there that you probably don't want to consume a large amount of. And it's not very practical, I think, to think about eating the seeds. It also would get fairly expensive if you were eating them as a form of sustenance rather than as a way of getting a little bit of sulforaphane.
So how do you consume? Because what we do is we get a handful and throw it into our salad. That's basically what we do with our broccoli sprouts. What's your routine with this?
I do that, certainly, but frankly, I also sometimes have a complete salad made of broccoli sprouts, or broccoli and I mix red clover, sometimes lentil sprouts in with them. So I have just a sprout salad, and I throw artichoke hearts, olives.
We actually do that too.
I've made it often with cherry tomatoes and onions, and cut very thin, and adding lots of sprouts. So, but what is an average amount that gives us what we need? And what is what we need? What is the amount that we need?
What's the dose?
The dose.
We've written papers on what's the dose, and the titles of those papers end with a question mark, and that's sort of the conclusion we come to. So the dose depends on a lot of things, but I would say for a decent rule of thumb is 2 to 4 ounces of broccoli sprouts. If you do buy them in the supermarket, which none of us do, it sounds like, it's one of those clamshells. that of sprouts, between a half and usually those are 3 or 4 ounces. So I probably, on days when I eat a salad of sprouts, I am sure I eat more like 6 or 8 ounces of them. But I think the comparison actually goes back not as much to animal studies and clinical studies, but it goes back to epidemiology. And the epidemiology of cruciferous vegetable consumption is very rich. It goes back to the mid 1970s at least, showing that people who ate in the sort of general conclusion that comes to mind is 2 servings per day of broccoli or of cruciferous vegetables led, in one of the early studies, to a 50% reduction in the risk of colorectal cancer. And those sorts of numbers have been echoed with breast cancer and prostate cancer and some others, in some cases with large cohort studies. So if you talk about 2 servings a day of broccoli, that's 200 grams, something like that. We actually looked at some of the epidemiologic data many years ago now and looked at broccoli consumption that other people had reported and agreed that if you divide consumers of broccoli into deciles, 10% increments, the lowest consumers, of course, ate none. The highest consumers ate something like a pound or a pound and a quarter of broccoli a day, which is huge. It's a lot. But so if you take that number and say, okay, at least some people eat that much. There are no adverse effects to eating broccoli except gas perhaps and maybe some indigestion, but people tolerate that much. And so if you extrapolate that number to broccoli sprouts, we've talked about there being let's say a 50-fold difference, 20 to 50-fold difference. I'll narrow that gap between sprouts and market stage. Then back calculating 2 to 4 ounces of sprouts is a reasonable number. That said, the numbers in terms of glucoraphanin itself, consumption in clinical studies range from about 30 milligrams per day of glucoraphanin to about 800. We did studies in China where we were looking at liver cancer prevention, biomarkers of liver cancer prevention, and biomarkers of air pollution detoxification. And we had as much as 600 to 800 milligrams of, we actually had as much as 600 to 800 micromoles per day of glucoraphanin, which translates to about half that number of milligrams per day. So, you can relate that back also to servings per day of broccoli or broccoli sprouts. And all of those clinical trials, I should say, were fully approved by multiple IRBs, ethics boards. And one of the questions that was always asked is that a reasonable amount of glucoraphanin to be getting? And so we always wound up having to go back to compare that number to what one would get if one was an enthusiastic broccoli consumer. So in other words, to go back to those epidemiologic studies and say, yeah, that's a reasonable amount. Is 5 times that too much to give as a pill, as a powder? We don't know, but we've not tested that because we saw efficacy, we saw effect at some of those higher concentrations. And I'd also just like to mention before I forget, more is not necessarily better. What we don't know, and what is very hard to get people to look at in a clinical study because of the risk involved that they won't find anything and then they won't get funded anymore. But we don't know how low you can go. We've done trials with autism, and we saw effect at the dose we gave, but frankly, we would like to to look at not just higher doses, but also lower doses, because the threshold for response we talked about many pathways that sulforaphane is involved with. Well, obviously, there's not an optimum concentration that's the same for each of those pathways. It's different. If you view it as a drug, which I don't like to do, it's a food, it's part of a food. But if you view it as a drug, you need different concentrations to do different things, right?
And so with autism is, of course, incredibly interesting because there are so many families and so many parents that would love to hear more about this. And there is some help, but the studies are very small so far. And they're given some benefits, but not the full benefits. And like you said, there are limitations in those studies in terms of dosage, in terms of delivery system. And we know that kids who are on the spectrum tend to have a lot of difficulties eating, sensory stuff. So you can't really give them the sprouts, most likely. You can't really give them enough broccoli to get enough sulforaphane to work with them. So now we go to supplements. And so supplements in general, even for people that can't or find it difficult to make the sprouts on a daily basis or to cook the broccoli on a daily basis. So what about supplements? Supplementing sulforaphane?
Yeah. So, to do these things as clinical studies for example, with disease interactions or looking for remission of disease symptoms is very difficult, because the minute you start talking about curing or preventing a disease, you're in territory that the FDA says no, forget about it for a supplement. If it's a drug trial, sure, you can spend many millions of dollars if you can find support for it. But again, we're talking about a natural product, a plant, a phytochemical, and we want to keep it that way. We don't want people to think of it as a drug. We want people to think, if I can help my symptoms, I can help reduce my symptoms of diabetes or of autism by eating differently, right? So, one of the things that we're actually a former doctoral student of mine who's now at Purdue is putting together a trial to try to use some sensory analysis and some flavor masking to create foods that are acceptable for children with autism who, as you say, have feeding aversions, eating aversions many times, so that we can do a trial using both broccoli sprouts and moringa with autism. So that's sort of going to very much the food side. But yes, for most of the trials we've done, we've had to look at what one would call a supplement, which is at its simplest, it's a freeze-dried extract of broccoli seeds or sprouts. And so in many of the clinical trials that I've done or been involved with, I have actually made, I had large batches of broccoli sprouts grown. I went out to the West Coast to the largest freeze dryer in the country, literally had them remodel their R&D floor. We boiled up 300 or 600 gallon kettles full of broccoli sprouts. Then we took that extract and piped it into freeze dryers and immediately dried it. So there's no funny business. There's nothing going on except extract and dry in a way that preserves the quality of the phytochemicals, or phytonutrients as some like to call them. And then we either put them in sachets or we had them put in capsules, in gel caps, with the idea that we'd be able to regulate the dose exquisitely. That way, you give 3 capsules, how many milligrams of glucoraphanin are in it. And B, unfortunately, with the idea that most Americans love to take pills, and they don't think anything's happening unless they take a pill. It's also easy to make a placebo when you do a clinical study that way. You give them another pill with colored powder in it. When we went to China to do our studies there, we, as I recall, well, I think one of the studies we did use pills or tablets, but with most of them, we made a beverage out of the broccoli sprout extract. Do you call that a supplement or do you call it food? We called it a food. In the U.S., when we did these studies with the extract that I handmade, we called it a supplement. Now there are very many supplements on the market with glucoraphanin. Well, I don't know very many, but probably 25 or 30 that can contain it or purport to contain it. So there's certainly a widespread availability of supplements to be used in clinical studies. Unfortunately, some of them are garbage, some of those supplements. And with full disclosure, I'm now consulting for the company that we started, Brassica Protection Products. And I know, for example, that any supplements that include their glucoraphanin have what they say they have in them, because I know the rigor with which the product is analyzed. But I mean, you can go on the internet and find all sorts of things that say sulforaphane cures cancer, and and here we're selling you some.
So supplements always contain glucoraphanin, not sulforaphane directly. Is that correct? Or glucoraphanin with myrosinase?
Correct. Those two. There are sulforaphane is very, very difficult to stabilize. It's very reactive. There are some supplements that claim they have sulforaphane. In most cases, I don't believe them at all. And we've had many of those analyzed. When I was at Hopkins, I wanted to put on our website, I wanted to put up a list and out the supplements that don't have what they claim they have in them, because we could do those analyses. The lawyers at Hopkins wouldn't let me do that for obvious reasons. But it's very hard to stabilize sulforaphane. And it's interesting when you think about the effects. When you take sulforaphane itself, it's absorbed pretty rapidly in the upper digestive tract, because it can be, and we don't know exactly where most of that absorption happens, but it doesn't wait till it gets down to the large intestine. With glucoraphanin as a delivery, most of the absorption happens after conversion by gut microbiota, so it's in the large intestine. And yes, there's less total available than when you give sulforaphane itself, but we really have no idea which is more effective if given on an equal molar basis. And then if you give a combination of glucoraphanin and myrosinase, some of that release of sulforaphane happens beyond the stomach, beyond the harsh acidity of the stomach. So in the upper small intestine. But it's a balance then between the supplied myrosinase and the myrosinase that are provided by your gut microbiome.
So an ingested myrosinase can survive the pH of the stomach, and
yes, there's a question of how active it's going to be once it gets through that gauntlet. And so one way to do that is to deliver it in tablets or in acid-resistant gel caps. We've seen what happens when some of those gel caps are filled with barium are swallowed, and then x-rays are taken. Sometimes they blow open in the stomach anyway, so it's not perfect, but yeah, that helps them. And we've actually,
as a cardiologist, one of my interests is, first of all, is there any data on direct cardiovascular benefits, but also with inflammation, because inflammation is a major fundamental cause of a lot of diseases, including heart disease. And I know that there's some significant and anti-inflammatory effects. So I just wanted to hear about that.
Yeah, so there are a number of studies looking at ischemia reperfusion, which speaks to the both cardiac ischemia reperfusion and I'm trying to think of where the other liver or kidney, I believe. So there are certainly studies showing protection there, where you have as I understand, you're the cardiologist, but you have a major oxidative stress upon reperfusion. So it's quite.
Clarify what that means, ischemic reperfusion. So
Giovanni has to clarify that.
Just,
So we're talking about block. When we build up plaque in the coronary arteries or other arteries in the body, it causes something called ischemia. That just means low blood flow to where the blood is needed. And when that's opened, that's reperfusion. When it's opened with a stent or when it's opened after a heart attack with a blood clot dissolving medicine. So you're saying that this can help the reperfusion injury. Is that correct?
Exactly. And Sandra, thanks for reminding us. We've got to be careful throwing around terms. We're not.
She always keeps us on track as far as that's concerned. So that's great.
Well, no one's trying to show off using those terms. It's just that that's the way we speak when we talk about the effect. But yeah, so indeed, protection against reperfusion injury. It's certainly been shown in animal models. I'm not sure if biomarkers of those injuries have been examined in humans. I can't remember. But in terms of inflammation, there's a dramatic effect, system-wide, really, but on inhibition or inhibition of the NF-kappa B pro-inflammatory pathway. This, to define that term, this is one of the important inflammatory pathways that is upregulated in various situations of infection and causes inflammation. So sulforaphane does have an effect in damping down that inflammation. And does that occur in cardiac tissue? I believe that's been shown in a mouse model or a rat model, but
it also affects heat shock proteins as an anti-inflammatory, and that may also be related to its effect on autism. Is that correct? The heat shock protein effect?
Yeah, exactly. So people have looked at the heat shock response in a number of, or been interested in it in a number of neurodegenerative and neurodevelopmental conditions, including autism, non-alcoholic fatty liver disease, NALFD, and what's it called, steatosis, hepatosis.
Yeah,
But yeah, in terms of autism, this is an interesting story. Our colleague Andy Zimmerman, who was at the Mass General and at Harvard, back in in 2006 or 7, he and colleagues published a paper where they were looking at the heat shock effect in they were looking at the effect of fever in autistic children. And it had been noted by clinicians for a long time that sometimes, not always, but sometimes kids with autism who got fevers, their symptoms were dramatically reduced, and whatever those symptoms might have been, or some of the symptoms. So Andy and his colleagues decided to codify that. They did a clinical study, and they showed that, yeah, indeed, something like 50% of people with autism, when they get a fever, those symptoms relapse or regress. And then when the fever's over, when it's resolved, they go back to the state they were in. So Andy, Dr. Zimmerman, came to us in the early 2011 or 12 and said, look, you guys, us at Hopkins, have shown that, and others have shown that sulforaphane affects the heat shock response in cell culture. People have looked at it in animal models. Why don't we do a study in which we see whether sulforaphane, because it can upregulate the heat shock response, is effective in reducing symptoms of autism? So we thought about it for about 5 seconds and said, whoa, let's. And so we did that study and published it in 2014. And indeed, sulforaphane had an effect on some of the symptoms in a sizable percentage of kids with autism. There was a significant effect during intervention with a broccoli sprout extract and compared to placebo, and then that effect was more or less resolved after a month of not taking broccoli sprout extract. Unfortunately, because of the way recruitment was handled in that study, we didn't have statistical power to determine whether the febrile effect, the fever effect, played into this. In other words, was it only the kids who responded to fever positively that had an effect of sulforaphane? So we were never able to do that. And I've been involved. Well, Dr. Zimmerman and my team, our team, did a follow-up study. Also couldn't reach conclusions about the heat shock effect. And then I've been part of a larger study in China where, again, there was no ability to tease that apart. Yet we still think it may be important.
Just again, the heat shock proteins are proteins that do a good job at eliminating those proteins that get broken down in the cells and they're not utilized anymore. And that is one of the reasons why we think that Alzheimer's, dementia, any neurodegenerative diseases occur, because the heat shock proteins don't work as well to detoxify the brain. So that's one of
the denatured proteins.
Correct. So,
Detoxify cellular debris. Yeah.
And that is one of the reasons why also sulforaphane might be good for dementia and
for all
people for even schizophrenia, for the cognitive deficits that happen.
We have colleagues and I at Hopkins did a small trial looking at schizophrenia and did not see an effect. I've been part of a trial in China where there was an effect, and there was a larger effect on people with more cognitive impairment. And so I believe that group is following up, looking at cognitive impairment as a standalone outcome. And I certainly, you brought up dementia. Alzheimer's is certainly something that we think, because of the accumulation of misfolded proteins, may be helped. There's not been a good trial on that. I have a fascinating short story, which I will deliberately not get into, even if there was a lot of time, but there's a retired chemist in France, in Grenoble, France, who has Parkinson's, and after he's in his early 80s now, but he contacted me and others in the field shortly after he got his diagnosis and wanted to start taking broccoli sprouts. He wound up making his own extract of broccoli seeds. As a scientist, he was titrating the dose, monitoring his symptoms. He got, there's no IRBs involved, no university participation, no money. But he got some pretty remarkable remission of his non-motor symptoms. Not the motor symptoms, but there are a lot of non-motor symptoms that people don't realize really plague people with Parkinson's. He then had a number of friends in his support group who tried the same thing, took exquisite records for, I forgot if it was 8, 10, 16 weeks, but quite a long time. And again, something like half of them had pretty remarkable remission of some of their symptoms. This is, to me, absolutely fascinating, and I would love to get involved in doing that study, that kind of study, but being able to look at blood biomarkers, being able to, have a placebo-controlled trial like that, It's bloody expensive. And of course, I'm retired now, so I'm not going to write grants to try to do that. But I mean, this is, I thought, one of the more exciting developments I'd seen in a long time in the clinical world. But it's, most of the peer-reviewed paper reading the scientific public is blind to it because it's not something they can really publish. It's just anecdotes, right? It's just anecdotes, but it's, one of those phenomenal types of anecdotes that screams out to you, we've got to do this in a more controlled way where it gets more credibility.
I may pick your brain more about that. I have a friend who's an expert on in the supplement industry, and he has Parkinson's himself, so he would be very interested in learning more about this. There are some things online that's been posted that have been posted about this so I'd be happy to connect with. Yeah. So one last topic for me is, one of our key interests is anti-aging, increasing health span. And I know there's been some work done on progeria, and Sandra has a relative, Tom Estelli, who's an expert on progeria. And
of course, yeah.
I don't know if you've come across Tom or Tom.
I've met him. I attended one of the progeria annual progeria meetings where he spoke. I've met him.
So and progeria, for people who are listening, is just an advanced aging genetic problem where you age very rapidly. So the study of progeria has led to insights on anti-aging. So I know there's been some work done with sulforaphane and progeria. So I just wanted to get your synopsis of that.
Yeah. So that's one of I won't call it a failure of mine, but one of the studies that I was less happy about the outcome of. So, we actually got a little bit of funding from the Progeria Research, is the Progeria Research Foundation, I believe. But we got some seed funding to do an in vitro trial, to do a study looking at the effects of sulforaphane on cell lines, primary cell lines from people with progeria, and we didn't find differences that were significant enough to write home about, so to speak.
No.
It didn't rise above the level of a least publishable unit. So we didn't publish anything about it. But I believe Mistelli did use sulforaphane in a trial, and then there's a woman, DJ. The first two letters of her name, I've forgotten exactly what her name is, but she published also on the use of sulforaphane, I believe. So I think there was certainly enough promise in the thought that sulforaphane might be effective in reversing some of the effects. Progerin is the protein that's involved, a nuclear protein, as I recall. We didn't have enough funding. It wasn't long-term enough. We didn't put enough effort into it, I suppose, and we didn't find anything worth writing home about. But yes, I think there's potential there. Of course, with progeria, since you mentioned it, it's a genetic disease, and it's a devastating disease because people with progeria have a life expectancy only into the teens, right? Late teens, maybe. There's some records. I mean, the people who've lived into their early 20s, I believe, but it's also an extremely rare disease, extremely rare. Maybe 400 people in this country have it, something like that. And my data is a few years old by now, but so it's very difficult to get funding to study it. And even the drug companies, there's not enough, as this is very crass and meant to be, but there's not enough money in it for them. So there hasn't been a lot of effort to develop drugs for progeria. There's been repurposing of old drugs from, with other targets to see if they work in progeria. So a natural product or plant-based approach to I don't think you're going to cure it because it is such a devastating genetic disease, but to prolong lifespan and enhance lifespan of those with progeria is certainly, I think, something reasonable to look for. The other thing is, it may actually tell us something about normal aging. How do we extend, not necessarily extend lifespan, but as you mentioned, extend health span for people who don't have progeria. So,
and that's why that research was interesting, because if we can protect that, that kind of severe disease, we might be able to protect the less severe disease or normal aging, and have to intervene that. And it was also interesting when we were talking, I was thinking about the pollutants and how sulforaphane can actually be helpful in detoxifying the body from that different pollutant. I was thinking, should all pregnant women take it, to help, that would be another interesting study, right? To see during pregnancy, breastfeeding, and start introducing it early on into baby's foods supply.
Yes, I absolutely. I think so. There's absolutely no reason not to, and there's every reason to do so. I'm actually working with a startup food company that's developing infant foods that are that contain broccoli sprouts, and the part of the rationale of this company is to expose infants very early on to a wide range, 50 or so fruits and vegetables, so that they get the tastes and the flavors, so that as they turn into functioning adults or kids who can choose their foods, they're going to go for vegetables and colorful food and appreciate the fragrances and colors.
And actually, that starts during pregnancy, because what we know is that babies, when they start to have their solid foods, they go more excitedly to the foods that the mother was eating during pregnancy. So they already have tasted that food. So reminding pregnant women to be really extremely careful what you eat during pregnancy, because your baby's going to love it.
We actually did a podcast about this, about how to introduce foods during pregnancy and after, and to introducing solid foods to infants. So I think that's extremely important. This startup company, what's the name of it?
Called Food Nerd.
No.
One word, Food Nerd. Yeah.
This information in that footnotes, and actually, if you can give it whatever information about seeds, the sprouting seeds that you would recommend.
We get our seeds from johnnyseeds.com. I don't know if you have a good place to get seeds from. Any information like that, we'd love to post it for our listeners.
Or in fact, when Paul first discovered sulforaphane, and then as we were doing our early trials, we got our seeds from Johnny's, because it was a variety from called Saga, SAGA, that Johnny's first provided to Paul, I think, back in the early 90s, and we were buying from Johnny's for quite a while. I've personally switched. So there's a variety. There's an old Italian landrace or open pollinated broccoli called Di Cicco, DECICCO. That's a good one in that it typically has pretty high sulforaphane content or glucoraphanin content, and it's relatively inexpensive because they don't use it to plant for groceries anymore. There's another one called Waltham 29 or something that's available actually on Amazon that sprouts really well and is organic, of course. So those are the three varieties that I've typically looked for. But I'll give you some information to put in the show notes. I have to ask you when I learned about your podcast and after I read your delightful book, it was a great book, by the way, I started on podcast 1, so I haven't gotten to the breastfeeding and kids food one. What number is it?
That's a few in there, maybe 10, 12 or something.
The early ones? Okay. Well, I must have missed it then. Since I don't commute anymore, I used to listen to podcasts while I commuted. Since I don't commute anymore, if there's too long a podcast, sometimes I just choose the short ones for short drives. But
yeah, we try to keep our podcast, or if we have a long podcast, we'll split it up into 30-minute segments, because I think that's the usual time frame.
Well, this was really delightful. This is such, like you said, you've been working on this for many, many years, 30 years, 25, 30 years. That's a long time. And we're still very excited about receiving this information.
And then you should be happy about how many people you've helped, because through us, we spread the word about sulforaphane. And I think that's a tremendous benefit to, people out there, because most people don't know this. So even today, with all the nutrition knowledge that's out there, sulforaphane is always a new topic for me and my patients, Sandra and her patients. So you're indirectly helping a lot of patients through many people.
And you helped us significantly. So thank you for your work, all the years of experience and hard work you've done, all your research.
You're very kind, and I'm happy to spread the word. I think we should also emphasize that it's the combination. In my mind, sure, there's a place for supplements. I know you had a good podcast on supplements, or at least one, because I listened to that one. But I am very much a proponent of getting your phytochemicals, your phytonutrients in food, in that complex matrix of fiber and vitamins, minerals, and not dozens, not hundreds, not even thousands, but probably millions of phytochemicals. I mean, there are a lot of estimates on how many phytochemicals there are in the plant world, but in any individual plant, there are certainly thousands of them, many of them beneficial.
And unfortunately, we're losing the amount of plants that we have available. And so we're losing the biodiversity, both in our gut and outside.
And also the soil is depleted. Our son is a permaculture farmer in North Carolina, and his main mission is to create excellent soil. We talk about organic. That just tells us about the pesticides. It doesn't tell us about the quality of the food. And he is, he has this farm. He's had it now for about 7 years. His soil is still not ready for prime time in terms of its nutritional content. And it's an important factor, because when we talk about phytonutrients in plants, they get their nutrition from soil. And so,
of course, and the other part of that equation is that plant breeders, not to this isn't their fault, but plant breeders have selected against phytochemical content inadvertently for the last century. I mean, they have been breeding for ease of harvest. They've been breeding for disease. Everybody breeds for disease resistance, but for many vegetables and fruit, I mean, they're breeding for increased sweetness, uniformity. Never are phytochemical content or specific phytochemicals the target. So we lose them over time. I'll have to give you, for the show notes, there's a link to, I'll have to give you a link to, there was a National Geographic article with I thought a fabulous pictogram, a full page diagram comparing plant diversity, fruit and vegetable diversity, 100 year ago to the present.
I think I would appreciate that. I think Scientific American did a study on that, but I would like to get the National Geographic one. That sounds great.
Great one pager, and it says it all. As you say, Sandra, we go from a lot of variability to very little, and this isn't micro. This isn't the our world is getting less diverse. This is our plates are getting less diverse, far, way less.
Well, significantly less, and that is a reflection of the microbiome. The less plants, so the less diversity in your microbiome. And
very true also, yeah.
appreciate what that means yet for our health.
And also, we don't know when we go to a supermarket what the quality of the soil is. So that's one of the goals of my son is to create a soil quality gauge, sort of like a number where you know what your more or less what the quality of the soil is.
Yeah.
But I agree with you 100%. You and I have talked about this, about the phytonutrient is so important. This is one of the premises of our book is it's the quality of the food that really matters more than exactly what you eat. It's there's no such thing as a superfood. It's really a superfood way of eating. The more you eat, the variety of foods, the greater it is that you're going to have.
And the synergistic effect of foods that we need to understand a little bit more about it.
We're all on the same page. I completely agree. Yeah.
Well, Jed, thank you very much. And hopefully sometime in the future we can do this again. But we really, really appreciate this. Our listeners are going to love this and get a lot of helpful, practical information.
Thank you for inviting me, and I look forward to hearing it when it's ready. And have a wonderful day, and take care of your dog. Now he's been very patient, or she.
Take care, Jed.
Thanks. Bye-bye.